So today I'm going to talk about microscopy. First I'm going to introduce the two terms that are relevant when talking about microscopy. The first one is magnification. And magnification is the ratio of the image produced by the microscope to its actual scale. So we generally obtain the magnification as an actual number. And that number is, as I have already identified, the eyepiece and the objective lens. As you can see in this particular picture of the microscope, the eyepiece says times 10, so the magnification is times 10, whereas the objective lens is also times 10. This means that the magnification, the overall magnification, would be 10 times 10, which would be 100 in this particular case. So to obtain the magnification, you simply have to multiply the eyepiece lens magnification and the objective magnification. There's also resolution, which is the ability to see adjacent objects as separate from each other. As we can see in this example, we have two pictures of the same organelle or cell, and on the first picture we can actually identify everything that's going on in there. Whereas in the other one, we don't exactly understand if it's one organism, several organisms joined up together, because the resolution of the first picture is much better than the resolution in the second picture. Now, there are three different types of microscopes that I'm going to talk about. The first one is the light microscope, and there's a picture in there that you can see of a light microscope. The light microscope basically works by having a light at the bottom, which is shined from downwards to up, that lights up the specimen or whatever you're trying to look at and allows you to see it better. Here we have an image of a spinach using light microscopy. Here we have an electron microscope, which is much more complicated than a light microscope. I won't go into details of how it works, but basically, since it uses electrons, it gives much clearer and better images. There are two types of electron microscopy. One of them is SEM, which is the first picture. It's a scanning electron image. It provides much more detailed imaging. In this case, we have a Pyralidae moth, which is a butterfly. As you can see, it's pretty detailed. And the second picture is a TEM, which is a transmission, I think. Which is a transmission electron microscope. It is also very good and much more detailed than the light microscope, but it doesn't provide 3D imaging such as scanning electron microscope. And in this example, we have a spinach chloroplast. We also have confocal microscopes. As you can see, it's pretty complicated, and I won't go into how it works exactly. But basically, there's a laser that kind of slices through a specimen. It doesn't actually slice through the specimen, so it's good for looking at living things, because the laser slices through it, providing a 3D image, but it doesn't cut or kill the actual thing. And it also allows for multi-labeling. So in this first picture, we have several cells and nuclei, and we can see that what is stained in blue is the nuclei. The cytoplasm is in red, actually the cytoskeleton, and the green dots are ribosomes, I'm pretty sure. So as you can see, you can tell different parts of an organism. The second picture is a larvae of a Drosophila, so a fruit fly, and it allows us to get a much better picture of living things without actually having to kill them. As we can see, the human eye can see up to around 100 micrometers. Actually, more than that. Actually, we can see around like 1 millimeter, or maybe like a tenth of a millimeter. We can see some eggs, some pond organisms that live in ponds. But to see beyond that, we need a light microscope or electron microscope. The light microscope can see up to maybe again a tenth or a fifth of 1 micrometer. So we can see bacteria, but it can't see large viruses. For that, we would need an electron microscope. And that's it. Thank you for watching. I hope this helped you, but if it didn't, let me know.